I think you're on.
Perfect.
Thank you so much, guys, for your talks. And now over to you.
Hello, and welcome to MedNews Week conference, where our goal is to bring you the most cutting-edge information from medicine's global leaders. I am Muskan Joshi, and I will be your host for today. Before we begin, I want to first acknowledge the great team that helps us make these events a reality. Our Our founders are two internationally recognized researchers, Dr. Jan Leifman and Dr. Chandler Park, who is also the president of Kentucky ASCO. Our chairs include Dr. Park and Dr. Leifman. Our associate directors include Madhuri Balasubramaniam, myself, Muskan Joshi, and Gayatri Menon. Our associate managers include Alexandra Van de Keeft, William Wilkerson, and Ahmed Aziz. Our educational committee includes Sean Jikevitz, Shubha Pawar, Chaurya Harshal, Reda Khan, Sri Vallabh Bheneni, Helena Koloma, Soumya Nadar, Rabab Huned, and Imad bin Zahid. We also would like to thank our partners, Medi, the global leader in medical video educational content, and the National Society of High School Scholars and Honor Society, founded by the Class Nobel of the Nobel Prize family. The moderator for today's event is Rabab. And now to introduce our wonderful keynote speaker, Dr. Seth Pollack. He is an internationally recognized leader physician in the field of sarcoma and has led multiple clinical trials to completion, shaping the current research and the therapy that we know of sarcomas today. He is also the recipient of multiple grants for his work in sarcoma and solid tumors from world-renowned organizations, including the National Institute of Health. He has also served as the advisory board member of multiple organizations such as the Desmoid Tumor Research Foundation and is the executive committee member for the National Leiomyosarcoma Foundation and is the member of various societies, including the Connective Tissue Oncology Society and the American Society of Clinical Oncology. He is also the recipient of numerous national and international awards, including the Alliance for Cancer Gene Therapy Fellow, the Sick Call Legacy Award at the Northwestern Sarcoma Foundation, and the V Foundation Translational Research Award, only to name a few. Dr. Pollack also currently serves as the senior editor of several internationally renowned journals, including Molecular Cancer Therapeutics and Cancer. With that, it is my greatest pleasure to welcome Dr. Pollack on today's session. Dr. Pollack, the stage is all yours.
Thank you so much. That's really, really great introduction. It's such an honor and a privilege to be here with you. You guys have such a incredible, diverse international audience, and it's really just a pleasure. I'm going to share my slides. And can you see it okay?
Yep, it's perfectly fine. It's visible.
Great. So you hit it on the head. I'm a sarcoma medical oncologist. I treat patients with sarcoma, and I'm really passionate about developing immunotherapies for sarcoma. And I'm really focused on how we can really bridge the bench to the bedside and really flesh out the translational component so that we can really figure out how to take the science and make it work in the clinic. And I think sarcoma is a really great place to do that for a variety of reasons. Some of these tumors have really unique immunobiologies that I think make them good examples and model diseases for figuring out how to make things work. And I hope you'll get some of that from the talk today. Sarcomas are a type of cancer. But they're not just one type of cancer. There's a lot of different types of sarcoma. The sarcomas comprise about 1% of all cancers, which sometimes people will poo-poo. They'll say, oh, that's not important. But actually, 1% of all cancers is a lot of people. So many people are affected by cancer. And in the US, 1% of all cancer is over 20,000 people a year. So that's a fair amount of patients. And we divide that up into the bone sarcomas. So you may have learned about osteosarcoma, Ewing sarcoma. Those are two sarcoma types that tend to affect kids and young adults. Those are both very sensitive to chemotherapy, and chemotherapy is a really critical part of the treatment paradigm for those diseases. Chondrosarcoma is another very important type of bone sarcoma. And then there's the soft tissue sarcoma, and what used to be just another type of soft tissue sarcoma only 20 years ago, today GIST is really very different in terms of its treatment paradigm. And we know that in most cases, GIST is driven by mutations in the KIT gene. And because of that, it can be very sensitive to imatinib, sunitinib, regorafenib, ripretinib. And now there are new agents that are very specifically targeting specific mutations, such as avapritinib, which specifically targets the exon 18 PGFR alpha mutations that can occur in GIST. Rhabdomyosarcoma are a group of sarcomas, and frequently in kids, they'll be embryonal or alveolar rhabdomyosarcoma, which have their own chemotherapy treatment paradigm. But today, I'm going to be focusing on the rest of the soft tissue sarcomas. Over 13,000 in the US patients per year, most sarcomas, which are soft tissue sarcomas, which is still a great variety of tumors, as we'll discuss. There's so many different types of sarcoma, it can be overwhelming. And this is from a paper that was focused on the ultra rare sarcomas. So you can see that a majority of soft tissue sarcomas have one of the most common subtypes. So undifferentiated pleomorphic sarcoma, a lot of us back when we were in school, we used to call those MFH, or malignant fibrous histiocytoma. Today, the nomenclature is different, and a majority of those old, very poorly differentiated sarcomas are called undifferentiated pleomorphic sarcomas. But there's a lot of little flavors within that, spindle cell sarcomas that can be poorly differentiated, and a lot of different types. Liposarcoma, another generally common type of sarcoma, and there's a few types of liposarcoma. Well-stroke dedifferentiated liposarcoma, which are driven by MDM2 and CDK4. Myxoid round cell liposarcoma, which we're going to talk about quite a bit, which is a translocation-driven sarcoma. Pleomorphic liposarcoma, which is a different type of liposarcoma. Leiomyosarcoma is another common type of sarcoma. These are cancers of the smooth muscle, and most people think, well, if it's cancer of the smooth muscle, it must come from the small intestine, right? That's where so much smooth muscle is, but that's not the case, right? So about a third of leiomyosarcomas are uterine, and then many of the other leiomyosarcomas originate in the smooth muscle that surround vascular walls, and especially in the retroperitoneum, in the smooth muscle surrounding some of the great vasculature. Like, people can develop leiomyosarcoma from the IVC, or from, it doesn't have to be from the IVC, it could be from smaller blood vessels. People can also get leiomyosarcoma in their skin or in their extremities in subcutaneous tissues. We'll also talk a bit today about synovial sarcoma, which is about 8% of sarcomas. It's another translocation-driven sarcoma. So you can see there's a lot of variety of sarcoma. These different tumors can be very different from one another. I mean, if you just imagine the difference between a liposarcoma and osteosarcoma, which we were just talking today, that's the difference between a cancer of the bone and the cancer of the fat. And they have very different biologies, very different treatment paradigms. But today I'm going to be focusing about the immunobiology of these tumors, which, as you'll see, varies quite a bit, just like their treatment paradigms do. And we're going to be talking today about cold tumors and hot tumors, which I think is a very reductive model of how we think of the immune system in cancer, but I think sometimes it can be helpful to think about things in this way. So cold tumors, of course, I know that you guys have had a lot of great speakers, some of the world's experts in immunotherapy, who I'm sure have brought up this concept before, but I'll just review that cold tumors tend to not have a lot of T cells. They frequently have a lot of immunosuppressing cells that could be like macrophages, which can have an M2 or immune inhibitory phenotype. They could have other immune suppressing cells like T regulatory cells, etc. They often have low expression of antigen presenting molecules like MHC. And sometimes they can have high levels of inhibitory cytokines that are just in the tumor. And this contrasts with hot tumors, which have a lot of T cells that tend to be exhausted in the tumor. And those tumors frequently will have high levels of PD-L1. They will also have immunosuppressive myeloid cells like macrophages or myeloid-derived suppressor cells. Those tumors actually are more likely to have regulatory T cells. The regulatory T cells are probably a bigger component of the inflamed tumors on average. So on the one hand, it used to be that, now we live in the age of immunotherapy. So it's easier to fight a hot tumor with a PD-1 inhibitor or CTLA-4 inhibitor or any of the checkpoint inhibitors, because those tumors already have exhausted T cells that are in the tumor. Presumably, those T cells are specific for the tumor. And if we could just figure out a way to turn them on, then they would kill the tumor. So, in theory, when you're talking about immunotherapy, these hot tumors are closer to being targets for immunotherapy than the cold tumors. Oh, sorry. So the three kind of things I want to talk about today is our efforts to define what a normal sarcoma immune microenvironment is. And then we'll talk about the problem of inhibitory macrophages, which is what we think the crux of the problem is for sarcoma immunotherapy. And then we'll talk about how we can change the microenvironment to facilitate cellular therapy in sarcoma. So what's a normal sarcoma microenvironment? And we'll also be talking a little bit about radiation and how that impacts it, because that's a normal part of sarcoma treatment. And we did a study to try and characterize the normal sarcoma microenvironment, and we looked at some of the most common sarcoma subtypes: the undifferentiated pleomorphic sarcomas, the liposarcomas, the leiomyosarcomas, and we looked at synovial sarcoma because that's it's a very as you'll see later, it's a hot topic for immunotherapy, and this included two sarcoma types that have complex genetics, frequently the undifferentiated pleomorphic sarcoma and leiomyosarcomas, and two with relatively simple genomes: the synovial sarcomas, which are translocation driven, and liposarcomas, which can either be dedifferentiated liposarcomas, which are driven by MDM2 and CDK4, or the myxoid round cell liposarcomas, which are translocation driven. And you can see, although this figure out is, I don't know why it's not coming out so clearly, but the undifferentiated pleomorphic sarcomas have significantly higher levels of PD-1 and PD-L1 by immunohistochemistry. So this was very clear from the start. There are some patients with synovial sarcoma that will have PD-1 or have intermediate PD-L1, and those tumors don't respond to PD-1 inhibitors. Even leiomyosarcoma, which does not respond to PD-1 inhibitors, can have some PD-1 or PD-L1 staining, but it's the undifferentiated pleomorphic sarcomas that unquestionably have higher levels of PD-L1 expression in the tumor. And we looked by nanostrain to characterize the immune-related gene expression in these tumors. And what we found is there was a striking separation among these tumors for genes related to antigen presentation and T cell infiltration. So of course, because I know there's all sorts of folks in this very large audience that you guys have. Just a reminder that when a T cell, like a CD8 T cell, sees a tumor, they really need to see antigen, and they don't just see antigen on the cell surface, they see antigen presented by MHC class 1, right? So, we were interested in characterizing that antigen, that antigen presentation, which we saw was different between these tumor types. And you can see that the two translocation-driven sarcomas, synovial sarcoma and myxoid round cell liposarcoma, have clearly less expression of the CD3D, which we used as a marker of T cell infiltration, and clearly less HLA-A, which is HLA and MHC are the same. HLA-A is one of the MHC related molecules, right? So they had significantly less, and the undifferentiated pleomorphic sarcomas had more when it comes to antigen presentation, and both the undifferentiated pleomorphic sarcomas and leiomyosarcomas on average had more T cells and more antigen presentation. And this is a bit tricky because the leiomyosarcomas don't seem to respond to checkpoint inhibitors just generally. They have a wide variety of expression of these things. But the undifferentiated pleomorphic sarcomas do, as we'll talk about in a minute. IL-7 receptor was also a marker for T cell infiltration, which was lower in the synovial sarcoma and myxoid round cell liposarcomas. And the same was true for HLA-DP, which is a class 2 MHC or HLA. So that this is kind of the tumors don't generally express class 2 MHC, but could be reflective of antigen presenting cells in the tumor microenvironment, myeloid cells, immune cells in the microenvironment. We also did TCR sequencing, and I'm going to talk about TCR sequencing quite a bit in this talk. So remember, back to your immunology days, when T cells differentiate, or when T cells form, during development, they undergo rearrangement of their T cell receptor, right? That's how you get such a incredible diversity of different T cells, right? Your T cell population in your body, you have all the T cells that you need right now to recognize whatever is going to come your way, be it cancer or infection. And the thymus, of course, during development will help to eliminate the autoreactive T cells. But because these T cells have undergone a gene rearrangement, you can do a sequencing to describe the T cell repertoire. So you can see over here, this is one of our samples. So on your lower left-hand corner, this is a TCR sequencing done on the Adaptive Biotechnologies platform. And we sequenced one sample, and you can see this has a number of different, it has lots of different TCRs in there, T cell receptors, TCRs. And the most common TCR that was in this sample comprised 0.27% of that sample. And you can see the amino acid sequence. And then the next one was 0.15, and you can see the amino acid for that one. And you can summarize this by a metric called clonality. And I've taken this graph in the middle here is a graph to illustrate the concept of clonality. It actually means something, and we're going to discuss it later on, but here I'm just going to use it to talk about the concept of clonality. So you can see here that in the dark red, this is an illustration of how much each T cell receptor is comprising the total one. So you can see the dark red is comprising the most common T cell receptor that was in this sample, and that makes up about 10% of the T cell receptors in a sample. By contrast, the top 10% over here is over 9,000 different T cell receptors. So that's the top of here. Now we did a treatment, and we'll talk about what the treatment was, and then this sample changed. So now the top TCR comprised almost 40% of the T cell receptors in the sample, and the TCR is 1,001 to 10,000, so 9,000 TCRs. The 9,000 lesser common TCRs comprised less than only a few percent of this sample. So this would be a sample where we would say the clonality is very high because it's skewed towards the most common T cell receptors. So we did this with our samples, and first of all, we characterized by using this technique, you can actually be very accurate in terms of quantitating the number of T cells that are in a sample. And you could see that the synovial sarcomas had a very low T cell fraction. The myxoid round cell liposarcomas also had a very low T cell fraction. And the undifferentiated pleomorphic sarcomas had a significantly higher T cell fraction. And this was true of the clonality as well. So the synovial sarcomas tended to have a low clonality, as did the myxoid round cell liposarcomas, as did the other liposarcomas. But the leiomyosarcomas had a little bit more, and the undifferentiated pleomorphic sarcomas had a significantly higher clonality. And what we think this means is we think this is reflective of the function of the T cells in there, that if your clonality is low, that it's more likely that the T cells are just randomly assorted there because there's no specific clones that have seen their antigen, have started to replicate, and have become a more dominant portion of the T cell repertoire in those tumors. Whereas the ones with a high clonality, there are some dominant clones that are probably specific for tumor antigens. And these results with regards to the tumor histologies are, at least with respect to undifferentiated pleomorphic sarcoma, it seems to be consistent to what we see in terms of response to PD-1 targeted therapy. So you could see here undifferentiated pleomorphic sarcoma in this landmark study, the SARC-28 study. 4 of the 8 patients treated with pembrolizumab here had durable responses to this pembrolizumab, a PD-1 inhibitor. And the dedifferentiated liposarcoma, which is somewhere in the middle, they had some patients with disease stability, maybe some responses. I mean, they did see 2 responses, but they were not as deep or durable as the undifferentiated pleomorphic sarcomas, but they had some durable disease stability. But no responses with leiomyosarcoma, 1 kind of very short-lived response in synovial sarcoma. I'm just going to emphasize the point right now that liposarcoma is 2 different diseases, right? The well-stroke dedifferentiated liposarcoma, and it's 3 diseases, but I'm going to be talking about 2, the 2 most common ones, right? Pleomorphic liposarcoma is an unusual one. There are people who are saying maybe there's more than 3 types, but I think there's kind of 3 types that everybody agrees with. And when I talk about well-stroke dedifferentiated liposarcoma, some people just have well-differentiated liposarcoma, and that generally has a good prognosis. The histology looks like normal fat, but if you have dedifferentiated disease, this is a very different tumor type. It's a very different tumor appearance histologically. So this patient has both a well-differentiated liposarcoma tumor, that's this dark one over here, and a dedifferentiated tumor over here, that's this lighter tumor. And the dedifferentiated liposarcomas, that's more of the aggressive portion. That's the portion that's going to really cause the patient problems. Myxoid round cell liposarcoma is called that because there's 2 components to it. There's a round cell component, which on this histology section is at the bottom, and the myxoid component, which is at the top. And if you're just seeing a patient for the first time, the round cell component is the component of the tumor that conveys a worse prognosis, that's associated with an aggressive clinical behavior. And you need to really watch out if your myxoid round cell liposarcoma patient has a big round cell component in their tumors. And we took a really careful look at the immune microenvironments in these liposarcomas, and we found that there were certain immune characteristics that were associated with a favorable outcome, with a recurrence-free survival that was over 3 years. And the big one was the presence of B cells. CD4 cells also made a difference. And then actually, the big one against this good survival was the macrophages and CD4 cells. or the macrophages and CD14 positive monocytes. CD4 cells were pushing things in the right direction. And we did this TCR sequencing for these dedifferentiated liposarcomas as well. And we found something really surprising, and it is something I don't know how to explain, something that my lab is really focused on right now, something that I think is a really fascinating question, is that when you looked at, so this is a chart where you map all of the different samples with respect to their T cell fraction, what percentage of the cells in the sample were T cells, and the T cell clonality, which is what we were just talking about before. And you could see that if you were in the upper left quadrant, we would think that probably that wouldn't be such a bad thing because, as I said before, a high clonality represents what we think is an antigen-specific response. But if you were in the upper left quadrant, it had a huge impact on your survival. So you could see that not in the upper left quadrant, lots of patients were living for many, many years after their resections, whereas not so if you had a T cell receptor profile that was in the upper left quadrant here. That's something that I don't understand and something that we're trying to sort out now. What about the macrophages? As I told you at the beginning, we think the macrophages are such a critical part of the tumor immune microenvironment in sarcomas. And you can see that this is, just because I know that not everybody does immunology all the time, just a reminder that macrophages have a lot of different functions, and they are an immune cell that is in charge, that plays a role in defending the host immunity, right? So they can present antigen. They can produce inflammatory cytokines like interferon gamma and TNF alpha, and they present antigen to T cells to stimulate the T cell response. And there are markers like TLRs, like CD8086, like MHC class 2, like INOS, that signal that this is an inflammatory macrophage. This is a macrophage that's there to improve immunity. Likewise, macrophages also play an important role in wound healing. And in the midst of a wound, it doesn't help anybody to have a big inflammatory response when you're trying to heal somewhere. And that's why, those macrophages are producing cytokines like IL-10 and that tend to be NTGF-beta, that tend to be immune inhibitory and prevent the T cell response. And those macrophages tend to express CD163, CD206, etc. So those are M2 or inhibitory macrophages. And we know that these macrophages are in a lot of different types of sarcoma. This was a great publication that looked at a lot of different types of sarcoma, characterized the macrophage infiltration patterns. You can see that most of these sarcoma types have a lot of macrophages, but especially in all the more common types of sarcoma that we've been talking about, the undifferentiated pleomorphic sarcomas, the liposarcomas, the leiomyosarcomas. Just a point that the leiomyosarcomas have a fair number of this CD163 positive macrophages. And we saw this in our data sets as well. Lots of different similar macrophages across all the different types that we looked at, but probably a little bit more in the liposarcomas, the undifferentiated pleomorphic sarcomas. And we looked at the immune infiltration in myxoid round cell liposarcomas and synovial sarcomas in particular, because I'll be talking about later, we're really interested in targeting these with immunotherapy. These are very immunologically quiet tumors. We've already mentioned that they have relatively low numbers of T cells. They have relatively low numbers of immune cells across the board, but they have a lot of macrophages relative to these other cells. And in leiomyosarcoma, it's been really well documented that if you have inhibitory macrophages, that they're especially if you could see these these two graphs at the bottom. This was seen not in uterine leiomyosarcoma, but in non-uterine leiomyosarcoma. There was a statistically significant worsening of outcomes when a patient had inhibitory macrophages that were seen on IHC. So one thing that we know, so we were really interested also in how radiation impacts this. And I'm going to come back to macrophages in a second, but we looked at patients with sarcoma getting neoadjuvant therapy, because a lot of these patients will get radiation. Most of these patients also had chemotherapy too, but we just as part of our standard clinical practice, we would collect a biopsy, then a patient would get neoadjuvant therapy, which almost always included radiation, and did include radiation here, sometimes included some chemotherapy as well, and then they would go for surgery. So what we did is we compared the immune microenvironment from the patients before their neoadjuvant therapy to after their neoadjuvant therapy. And you can see we had a whole different variety of different sarcoma types, a diversity in sarcoma sizes, grade, mainly intermediate or high grade, but a lot of different types of sarcomas. And what we saw is a big increase in the antigen presentation molecules like HLA-DR. We saw a big increase in B cells and natural killer cells, and we saw an upregulation of genes related to antigen presentation. And we saw a big increase in macrophage-related markers. So on the one hand, while radiation might be killing the tumor and thus is absolutely essential for improving outcomes of these patients, we know that that's incontrovertible with excellent data, we may also be increasing the inhibitory macrophage population in these tumors with radiation. We thought that maybe radiation would be sensitizing these tumors by just increasing the T cell infiltration and increasing the antigen presentation, which it does, but it also has a big impact on the M2 macrophages, as you can see by the increase in CD163, the increase in CD206, etc. So how can we overcome this problem of inhibitory macrophages in the tumor microenvironment? Well, one thing we were really interested in is whether we could use a toll-like receptor to alter the microenvironment. We know, so toll-like receptors are part of the innate immune system. And when I say innate immune system, again, for those of you, because I know there's so many people watching, if it's been a while since you did your immunology, we talk about the innate immune system in contrast to the adaptive immune system. The adaptive immune system are the cells like the B cells and T cells that have super specific receptors that are going to attack a given pathogen, like a T cell that's going to be specific for a virus, or a B cell that's going to be produce a specific antibody. Toll-like receptor is part of the innate immune system because it's there to activate the immune system, and it's fixed. And in whatever context that you have, the cells have toll-like receptors that the immune cells will have toll-like receptors that are potentially activating, and toll-like receptors are found in pathogens. So these are signals that the immune system has developed to develop specific pathogens. And we were interested, we decided to use a toll-like 4 receptor agonist, and this one's specific, called GLA, is from the bacterial cell wall. So it's a component of the bacterial cell wall that we injected into tumors, and we did it concurrently with radiation to see if we could reverse this macrophage change that we had observed in this prior study, to see if we could make the macrophages in those tumors become activating, because that's very well established that toll-like receptors can change inhibitory macrophages to activating macrophages. And this was a phase 1 trial. So because it was an experimental trial, we did this on patients who had metastatic disease, but had an indication for radiation. So they had metastatic disease, but an injectable tumor that was causing them pain and discomfort, and which the tumor really necessitated, or at least the patient wanted radiation to help alleviate their symptoms. And the tumors that were injected did really well. None of these tumors grew significantly, A majority of them had nice responses, at least some tumor shrinkage or a response that was significant. As I said before, we had a wide variety of tumor types. So this was a small study, so we couldn't really pick out which sarcoma subtype was having the benefit. And in some of the patients, because these were patients with metastatic disease, we were able to follow multiple tumors. So some of the patients had tumors that were radiated and had the injection, had radiation, but were in places where it wasn't safe to inject, and had tumors that were not radiated or injected. And you could see the patients like that, the tumors treated with both radiation and the toll-like receptor did better, right? So you could see they had a deeper response compared with just the radiated tumor, and clearly the unradiated or injected tumors did worse. And this graph over here illustrates the tumor size to kind of hammer that point home. And you can see these CT scans that really show this very clearly. One question that people have been asking is whether these tumors themselves have toll-like receptor 4, and they do sometimes, but that didn't seem to be related to the response. Of course, there's a small cohort, but this study was modeled a little bit after some of the studies in hematologic malignancies, especially in lymphoma. There's a great researcher and scientist, Ron Levy from Stanford, who's done a lot of work with TLR9 and lymphoma. And in that case, the lymphoma cells themselves have TLR9, but that's not what we were trying to do here. Here we're trying to activate the immune cells that are in the tumor. Of course, there were some tumors that had a lot of TLR4, but that didn't seem to correlate with response. So you can see here are some of the best responses that we saw. One of them had very little staining of the tumor cells. They had immune cells in their tumor that stained positive, but the tumor cells did not look like they were staining for TLR4, whereas some of the other tumors did have some staining in the tumor. In the patients who had nice local responses, they had a big increase in the T cells, and they had an increase in the clonality as well. And we'll talk a little bit more about that as well. Here's some nice some MIHC staining that is showing the increase in cellularity, but also the increase in CD4 and CD8 staining that we see following the TLR4 injections along with radiation. Now, let's talk about clonality. So this is the graph that I showed you about before. This is in one of our patients who had one of the best responses. They had a huge increase in the clonality of their T cells. You can see here. And what I find, so here we're illustrating it, and you can see some of the specific clones that developed on treatments. This is from TIL that my lab is very good at growing out TIL, at least for analysis from these tumors. And you can see one thing that's so interesting to me, and I'm really interested, maybe some of you guys have ideas out there, and let me know. One thing that's just so interesting is that these T cells that were some of the biggest expanded T cells, they weren't just one single clone. What we saw is that there were a variety of gene rearrangements that resulted in that clone. Likewise, they didn't, you have different V beta gene families that can be cut up and comprise a T cell receptor. We saw different V gene families that comprise these T cell receptors, which is just so wild to me. And I don't know that that it's been seen in these sort of expanded clones before. Of course, we know that these sorts of TCR clones exist, but in the context of a cancer treatment, I think this is really something that's unique. And I'm really interested in these sorts of T cell receptors because I think that in my opinion, there's something about the fact that you have so many gene rearrangements converging on the same T cell receptor that they must be important, right? They must be for something specific and some sort of antigen that that is such that the immune system is primed to hone in on it. When we did single cell sequencing, we used a single cell platform that allowed us to really focus in on some of these clones. We found that they had a Th1 phenotype. These clones that were very expanded. So these were CD4 cells, and they had a Th1 phenotype based on the single cell sequencing that we did. We also looked at the PBMC clonality in these, and we saw that there was an increase in the PBMC clonality in the patients who had nice responses, and that a lot of the overlap in the expanded in the expanded clones in the PBMC had a lot of overlap with the pretreatment TIL. So I'm really excited about these results, and I think that there's going to be a lot of ways that we can apply it. And maybe in the question and answer session later, we could talk about kind of how we can kind of exploit some of these findings. But TLR4 is not something that you can just inject IV. It's too dangerous. It's too inflammatory. So we're also really interested in how we can manipulate macrophage phenotype using systemic treatments. And one of the things that we've been interested in is trabectedin. And trabectedin is a very old drug. Now it's was developed sort of right in the wake of Taxol. So Taxol was discovered. Of course, Taxol is the chemotherapy that comes from the yew tree. And after Taxol was discovered, there was a sort of a gold rush of people looking at natural compounds to see if any of them had anti-tumor activity. And trabectedin was discovered in a sea urchin through a high throughput assay. So basically they looked at the sea urchin, they looked at all the different compounds in the sea urchin, and they tested all of them against cancer, and they came up with trabectedin as an effective agent. And they didn't really know how it works at first. And through additional studies, we found that it binds in the DNA's groove and can prevent cell division through that. But we found that it's been shown that trabectedin is also very toxic to macrophages. So it has probably a primary effect, which is directly on tumor cells, but it also binds and prevent to macrophage DNA and prevents and kills the macrophages that are in the tumor. And that's probably an important part of the effect. So this is a great Cell paper where they looked at tumors and they found that, so this is a tumor. If you look at figure B here, the untreated tumor grows a lot. If you treat with trabectedin, that kills a lot of the tumor and you get the red curve. But if you use trabectedin and you also infuse this mouse tumor with the macrophages, you reduce a lot of the efficacy of the trabectedin. So like half of the effect in this mouse model of the trabectedin is coming from killing the macrophages, right? So there's some effect still. The trabectedin is acting on the tumor, but some of the effect is from killing the macrophages. And we looked at, so we did sequencing of tumors that were in patients that were treated with trabectedin in our institution. And we found that if you had a gene signature with the patients who had this M2 gene signature did much better than the patients who had this other macrophage gene signature. And the same thing with myeloid-derived suppressor cells, suggesting that there's a really important link between the myeloid cells and their response to trabectedin. in our patients. So we combined trabectedin with this PD-L1 inhibitor avelumab in a phase 1/2 trial in liposarcoma and leiomyosarcoma. And my philosophy is that it would probably be a good combination, maybe even a better combination in some other tumor types, but liposarcoma and leiomyosarcoma are the two tumor types in the US where trabectedin is FDA approved. So we wanted the on-label indication for trabectedin. And there was a dose escalation and then a phase 2, a wide variety of patients within these two tumor types. And patients could have had a variety of different lines of therapy, up to 5 lines of therapy. So some of these patients were quite refractory. And we had a number of patients with really, really durable responses. And I thought this was really encouraging. Now, the primary endpoint for the study was response rate, and we didn't quite meet the response rate endpoint, but which I think is unfortunate, because I would look at a patient like this patient that's going out beyond 2 years, these couple of patients in blue who had stable disease, but these are certainly successes because these are aggressive cancers. They tend to have overall survival of less than 2 years in the metastatic setting. So if you have a patients that are doing this, that's a success. And we saw a 6-month progression-free survival of over 50%. So by some metrics, this was a really effective combination, even if it didn't meet the primary endpoint. And that's why I do hope there are future efforts to, and I think probably there are some additional things, and we could talk about this too, maybe to even make this combination even more effective. And when we did TCR sequencing of these patients, PBMC, so this is pretreatment PBMC, the patients that responded better to trabectedin had who had partial responses had so this is pre and post treatment, had higher clonalities, both pre and post treatment. So if you had a partial response, you were unlikely to have a low clonality. So, you know, they didn't quite not overlap. So you could still have an intermediate partial clonality and have a partial response. But these patients who responded had higher clonalities, suggesting a T cell repertoire. Remember, this is in peripheral blood now, but that their peripheral blood T cell repertoire that's skewed towards a specificity against tumor antigens. There's also a large body of literature showing that doxorubicin can be an immunosensitizer. And I'm talking about it here because, you know, one of the reasons why we got interested in doxorubicin was because it kills macrophages. And we did a number of experiments looking at the tumor immune microenvironment in various models and showing that doxorubicin did a good job killing the macrophages without having a big impact on the T cells. But even before the age of immunotherapy, people were using doxorubicin to kill tumor and cause antigen presentation. So that's one reason why we think that doxorubicin can be a really great addition to immunotherapy. And of course, in sarcoma, doxorubicin is the standard frontline agent for metastatic soft tissue sarcoma. And that is despite the fact that it really isn't a good drug. So you can see here, it's the frontline agent. There's nobody who's questioning that. I'm certainly not questioning that. Some people will say that you should use a more aggressive regimen with doxorubicin and ifosfamide, and that does give you a better PFS, but the overall survival is probably very similar, and it's terrible quality of life while you're on the combination. So at most institutions in the United States, if you have widely metastatic sarcoma and there's no thought about resecting oligometastatic disease, et cetera, you're going to get single agent doxorubicin. And you can see the PFS for single agent doxorubicin is not what we want. It's 6 months or less. And the response rate is not what we want. It's 20% or less. This is on all of the studies, not all of the studies, but some of the major studies testing doxorubicin versus doxorubicin plus another agent. So we did a study of doxorubicin plus pembrolizumab, the PD-1 inhibitor, and we saw better PFS than had been seen on these prior studies, 8.1 months. We saw better overall survival, 27.1 months, and we saw some patients with very durable responses. You could see this going out more than a year with either stable disease or responses. And I've talked to some of these patients now who are 5 years out, who still haven't progressed with either dedifferentiated liposarcoma or undifferentiated pleomorphic sarcoma. So I think this is a really good regimen for some patients. And you could see in, if we just look at the undifferentiated pleomorphic sarcoma patients, these CT scans illustrate the fact that not all the tumor shrinkage is just coming from doxorubicin. So you can see here, after, this is a UPS tumor. After doxorubicin, it shrank, but the shrinking continued after 6 months of doxorubicin. Same thing with this dedifferentiated liposarcoma. And you can see that in each case, there were some patients who progressed, but 2 out of the 3 undifferentiated pleomorphic sarcomas had very durable responses. Half the dedifferentiated liposarcoma patients had durable responses. And this has since been kind of reproduced in another study from North Carolina. So we have now we have an upcoming big multi-center randomized trial that's we're finalizing the protocol with ECOG Akron. It's been NCI approved. It says under review at the NCI. The NCI has approved it, CTES has approved it, but the final protocol is still being tweaked. But I expect it'll be out in the next couple months. And this is going to be a big randomized trial throughout the US. And there's discussions that maybe even potentially elsewhere, looking at 160 patients randomized to either doxorubicin and pembrolizumab or doxorubicin alone. And I'm excited about this because I think it has the potential to change the standard of care. So let's talk about cellular therapy. This is one of my passions. This is kind of how I got into the field of immunotherapy of sarcoma. I was very interested in targeting some of these tumors with that express high levels of cancer testis antigens. So again, you guys have had some amazing speakers in this group. You may have heard about cancer testis antigens before. Cancer testis antigens, or sometimes they're called oncofetal antigens. That's like a new terminology for it, but they have a high level of expression in the testis. So this is the seminal vesicle. These are the precursors to sperm. So you have mature sperm in the middle of the seminal vesicle, the immature sperm at the outset, and the immature sperm express high levels of cancer testis antigens. There's also fetal organs like fetal kidney that will express, and fetal ovary that will express high levels of some of these cancer testis antigens. But most, and they can be expressed by placenta as well, but healthy tissues other than the testis and placenta in kids or adults do not express these. So there are good targets for cancer therapies. Some of the famous ones are NY-ESO-1, that's CTAG1B here. And you can see that the synovial, we looked at a variety of different sarcomas using a panel that we made with a lot of different cancer testis antigens. These, you can see a lot of them express MAGE-A9, a lot of them express MAGE-A4, a lot of them express PRAME, a lot of them express NY-ESO-1. So we were very interested in targeting these. Synovial sarcoma had already been described to express NY-ESO-1 when we did this study. But we were really excited that when we took a deeper look at myxoid round cell liposarcoma, every case that we looked at had some expression of NY-ESO-1. Since then, I think there are very rare patients that won't have any NY-ESO-1 or MAGE family antigens, but it's very unusual that they don't have even a little bit. And in most cases, it's homogeneous. So both with synovial sarcoma, in this study, over 70% of the cases, both synovial sarcoma and myxoid round cell liposarcoma generally have homogeneous expression of NY-ESO-1 and myxoid round cell liposarcoma and other cancer testis antigens in both synovial sarcoma and myxoid round cell liposarcoma. So I think there's been a lot of talk, and people get very confused about CAR T cells and gene-engineered TCR T cells. So remember, a CAR T cell, which is what's FDA approved for leukemia, ALL, lymphoma, these can be used for. They can be effective for CLL. They're very effective, these CD19 specific CAR T cells. And there are, of course, there's also CD20 specific CAR T cells. There's the BCMA CAR T cells and multiple myeloma. There's a lot of CAR T cells out there now, which are either approved or have some evidence of efficacy and are heading towards approval. These have an scFv, so that's like the part of an antibody that recognizes its target, and it's attached to a co-stimulatory molecule like CD28 or 4-1BB, and it's attached to like a CD3 zeta chain, which is what actually stimulates the T cell. And that will, when that recognizes a cell surface protein, the T cell will kill it. A TCR recognizes an intracellular protein that gets expressed on the cell surface by MHC. So you can see, and because of that, you have to have a specific MHC molecule, or a specific, in other words, you have to have a specific HLA type. And in the US, the most common HLA type is HLA A0201, which is why if you're going to try and target a patient with synovial sarcoma using a TCR, we require those patients to have HLA A0201, but there's no reason why you can't develop a TCR for HLA A24, which is the most common HLA type in some parts of the world, or any of the other class 1 HLAs, or even class 2 HLAs, right? And we've made TCRs for a lot of those, and there's companies out there that have TCRs against different HLA types, but it's a big production to make these T cells. Is very expensive for companies doing clinical grade production. It can be hundreds of thousands of dollars just to make one T cell product. That's not counting all of the costs that go into just treating the patient and your clinical research coordinators and getting the samples and doing the analysis to make sure that the T cells stay around, right? It's a big production. So far this has only really been looked at well in patients with HLA A0201. When I got interested in this, what I've always kind of been interested in is what the T cell repertoire is in the patient, and how can we make that more robust. So what I was doing is I was actually culturing out NY-ESO-1 specific T cells from these patients. So we would take a patient's blood, and you could see we use a reagent here that marks the NY-ESO-1 specific T cells in HLA-A0201 patients. And you can see this patient, you basically can't detect them in an untreated sample, but we would culture out dendritic cells, and we would pulse those dendritic cells using an NY-ESO-1 peptide. And then we would get a really nice population of NY-ESO-1 specific T cells, and we would use those to stimulate the patient's blood, and we would get, and that's what we would get, and then we would sort out those antigen specific cells, and we get a very purified NY-ESO-1 specific T cell product, and we would use those to treat the patients. And so we did a pilot with just these cyclophosphamide, with just these T cells using cyclophosphamide conditioning, and all these patients had some evidence of clinical benefit. One patient had stable disease for a year. One patient had an amazing reduction in the size of a liver lesion. One patient had some lung lesions that got smaller, but all the patients ultimately progressed. So they all had some tumor shrinkage, but they all had ultimate progression, which is kind of what's been seen on, I mean, some patients have durable, deep responses with adoptive cellular therapy. That's what you want. But most patients don't. Most patients will, even if they respond, even if they have a nice response, they'll ultimately progress. And that's really kind of the crux of the issue. How can we get these patients who have an initial response, how can we get them to have that response continue and be very deep? So you can see this is that liver lesion that I mentioned that had a nice response. This is a patient who went for resection of their lung lesions after they had adoptive cellular therapy and progressed. And this is staining for NY-ESO-1. And what you can see is that the NY-ESO-1 is still there. So they didn't fail because they lost antigen. So that's kind of something that can happen. Like in melanoma, there's lots of cases, not typical. I think it's overestimated how big an impact this effect has. But sometimes a patient will have, they'll be treated with MART1 specific cells and they'll lose MART1 expression in the T cells. That's called an antigen loss variant. Most of the time, that's not what's happening, right? Most of the time, we don't know why these patients progress. Also, one of the big determinants of success in cellular therapy is the persistence of the T cells. So you could see these patients had persisting T cells, and you can see this is by flow. They still had a nice, though not big, they didn't have a big population, but they still had detectable T cells in their blood. So how is it that they still have a tumor that's expressing a lot of antigen and NY-ESO-1 specific T cells in their blood, and it's not eliminating it? And this is what drives me crazy, because I think that we act sometimes like we know, like, oh, we just need to have more T cells in the blood. Oh, their antigens are disappearing. They're not. That's not what's going on. It's something more than that. It's deeper than that. We need to figure it out. One thing that probably is a problem is that the phenotype of these cells that are adoptively transferred changes in over time while they're in the patient. So what we found is that so we did a bunch of sequencing and analysis of these cells that I'm not going to show. They don't have an exhausted phenotype, like they don't express PD-1, they don't express LAG3 or TIM3, but they don't also have markers of activation. They don't seem to be producing a lot of cytokines, and you can see that when we treat them with IL-15, they if you just have IL-2 in there, they don't grow very much. But if you use IL-15, they have a massive expansion. They also start to produce a lot more cytokines. And when we used an organoid model, these IL-15 treated T cells just totally eliminated tumors in the organoid model. These are T cells that we took from patients who had progressed after adoptive cellular therapy. So we took patients' blood, either from our trial, or we also took them from patients on a trial at UCLA or at the NIH. And we took their blood and we co-cultured it with tumor cells in an organoid model. And without IL-15, with just IL-2, the tumor cells, which are green here, still grew, and the T cells didn't do much. But if we used IL-15, the tumor cells were eliminated. So the other big problem, one big problem that we see with these tumors is they have low HLA expression, low MHC, and low T cells in there. So what we thought that one drug that is very old drug that we know can sometimes increase MHC and can sometimes increase T cells in tumors is interferon gamma. That's a very old drug, was tested in cancer for many years, never really worked in cancer, although there are some important clinical places where it works. Chronic granulomatous disease is a terrible immunodeficiency that children can sometimes be born with, and it's a drug for that. There's a condition called osteopetrosis that interferon gamma can use. So we were able to buy it. And now it's very expensive, but at the time, it was actually owned by a different company and was very affordable to purchase for a clinical trial. So we did a pilot trial. This is a phase 0 study where we did a biopsy of some of these tumors. We treated the patient with interferon gamma, and then we did a biopsy after 4 weeks. So it's a phase 0 study. A phase 0 study means that the primary endpoint of the study was a biomarker. So the primary endpoint was MHC class 1 expression. That doesn't mean that you're not trying to treat the patients, just like in a phase 1 trial where the primary endpoint is safety. You still want efficacy, but the primary thing you're looking at is the safety, right? Here, phase 0 trial, the primary thing you're looking at is a biomarker. So it's not safety, it's not efficacy. You want safety, you want efficacy, but the primary thing that you're looking at is a biomarker. So we're looking at class 1 expression. We can see here, at least in this patient, their tumor expressed a lot of class 1 after the treatment. Of course, that was not typical. So this is the patients. You could see more than half of them had a big increase in their class 1 expression. Some of them, now these ones that don't look like they had a lot of class 1 expression, we still think this may be important and relevant because they went from basically nothing to having something. So if you go from 0 or basically undetectable to having 1%, I'm not sure that that is nothing, right? So 1%, of course, is not the same as 100% or over 80%, but 1% is still more than nothing. And this is a nice immunofluorescence. This is just gene expression on the tumors at the time of biopsy, showing that they had a profile more consistent with antigen processing presentation in the post-treatment sample. We also looked at NY-ESO-1 specific T cell responses in the peripheral blood. And we did find in 2 of the patients, we were able to show that they had a development of an NY-ESO-1 T cell response. So you can see here, these are PBMC treated with the negative control, whereas patients, the pre-treatment is on the left and post-treatment is on the right, and the post-treatment after treatment with NY-ESO-1 has a big NY-ESO-1 response. But we also saw an increase in PD-L1, particularly in the myeloid cells. So some of the patients had an increase in their tumor, and this is a striking one, but what we saw very consistently was an increase in the myeloid cells. So we had a trial of pembrolizumab, PD-1 inhibitor, and interferon gamma, but we did not see responses there. And we have some theories why now. So we're analyzing the samples in the lab right now, and I think we do have some clear answers for why this might be happening, and hopefully we'll be able to let you know about that in the coming year. We also tried interferon gamma along with the cellular therapy. So we gave interferon gamma for 2 days before our NY-ESO-1 specific T cells. We gave cyclophosphamide and then the T cell infusion, and then 2 doses of interferon afterwards. You can see this patient had a nice response in their lung lesion, but they ultimately, this is after the cell therapy, they developed this forehead lesion. So they did have technical progression, even though they were actually feeling a lot better after the treatment. So their pain kind of went away. They had shortness of breath, but that went away. They were able to go back to work. But this tumor was removed, and you can see it. This is post-treatment. This is the lung tumor on the right, on the left under D, and 10 weeks after is the forehead tumor on the right. And you can see NY-ESO-1 is still expressed, and E is the class 1 MHC, and that was not expressed afterwards. So there's definitely still more to do to figure out how to get interferon gamma to help these patients on cellular therapy. And 2, the second patient had a terrible, tragic myocarditis that happened, and this forced us to stop the study. Although I will say this patient had very advanced synovial sarcoma. So this was a patient who had a terrible prognosis. They were treatment refractory. They were in bad shape, regardless of what we did. But it was so tragic because I mean, it was such a wonderful person with such a wonderful family, and it made me think that we really need to come up with some better ways to develop cellular therapy for these tumors. And one thing, and you can see now that this is only going to become a bigger part of the conversation. So MAGE-4 TCR is probably going to be FDA approved in the US. There are a lot of patients who have durable either responses or disease stability, but most patients you can see either have a response that's short-lived or who progress. So it's an amazing therapy. I'm so happy it's here. It's going to totally revolutionize our treatment for synovial sarcoma, but there's a long way to go in terms of making it everything that it could be. So the thing that we're really excited right now is the most common solid tumor in pet dogs is sarcoma, and there's a lot of really good data that the sarcomas that the pet dogs get are very similar to the sarcomas that humans get. For example, they get osteosarcoma, and their osteosarcoma is very similar to human osteosarcoma. They get what's called hemangiosarcoma, which is very similar to angiosarcoma. They get what's called soft tissue sarcoma, which is very similar to what humans would have as undifferentiated pleomorphic sarcoma. And all of these sarcomas express a protein called B7H3, which is a very popular target for CAR T-cell therapy. There's a number of trials around the country targeting B7-H3 with CARs. There's a company that makes a monoclonal antibody that's been tested in the clinic. So we made a canine CAR T-cell using canine 41BB and canine CD3 zeta, along with B7-H3 SCFB. And it's in right now. We've treated several dogs at the University of Illinois, something that's we're very excited about. And this, I think, is going to really give us it's not a TCR, so it's a different thing, but it's going to give us a way that we can try and improve cellular therapy using a really, because when you treat mice, the way, if you're making gene engineered therapy in a way where you're being tumor specific, and you're using real human tumor, then you have to be using a xenograft, and the xenograft does not have a human immune system. If you use a fully murine system, then you're losing some of the human element. But a canine tumor has a complex immune system that's much more similar to human compared with the mouse, and has a naturally developed tumor, right, that evolved through all the same steps that a naturally developed human cancer goes through. So we think this is a great system to develop CAR T cells. And this is Tim Fan, who we're doing this trial with in CAR T cells. Jeff Bryan is a veterinarian from the University of Missouri, and we're testing out some intratumoral injection therapies, specifically using IL-12 producing treatment. So hopefully that will even further increase what we can do here. This is my lab, but there's a lot of other folks that aren't here. This doesn't include my clinical research group, who are amazing. Our great team of sarcoma surgeons and clinicians here at Northwestern, who are just fantastic. Some of my great collaborators, especially Stan Riddell from the Fred Hutch, which is where I used to be, Robin Jones, who also used to be there and is now at the Royal Marsden. And I really appreciate your having the opportunity to talk like this with you guys, and very happy. I don't know how much time we have left, but happy to take any questions.
Thank you so much, Dr. Pollack, for that wonderful, wonderful presentation. I have learned so much, and I want to tremendously thank you for such a wonderful talk and a wonderful presentation. So thank you so much for that. So now I will be passing on the mic to our moderator, Rabab.
Who's the director of Sarcoma program at Lurie Cancer Center of Northwestern University. We have received a lot of questions, and we'll go through them within the time. So for the first question, I'll be asking to you, Doctor. How do you think artificial intelligence has changed the therapeutic approach of sarcomas? And if not yet established, how far can artificial intelligence help in the future?
So the first part of the question is, I don't think it's had an impact yet, but I think it will. I think it's going to be huge. I think it's going to totally revolutionize how we deal with every cancer. So first of all, just in terms of the classification of sarcomas, you saw that pie chart at the beginning and how complicated the classifications of sarcomas are, but there's still types like the undifferentiated pleomorphic sarcomas. I'm not convinced that's even one tumor type. There could be a few different tumor types that are just stuffed in there together, and we're just not seeing how they're different. And I think artificial intelligence is going to really help the pathologists to sort through these tumors and figure out what's what. And by doing that, we're going to have better biomarkers for how to treat these patients, and knowing what's going to work for some patients and what's going to work for other patients. Likewise, with I don't know how much you guys have been hearing about radiomics, but radiomics is a way where you use non-intuitive features of the radiographic imaging. So things that you wouldn't normally see, a radiologist wouldn't be able to observe, in the images, I think artificial intelligence is going to help us sort through that. So that's kind of like low hanging fruit. Then in terms of things like I was showing you before about the T cell repertoire, right? I think artificial intelligence is going to help us sort through those data sets and really kind of identify patterns that we wouldn't have been able to see and find patients who are right for various therapies, find TCRs that are specific for tumors that we might not have been able to recognize as being important. And then, of course, in the gene expression, I think artificial intelligence is going to have a great ability to improve our ability to interpret things. But, one thing that artificial intelligence really needs to work well is the samples, right? So it'll have pathology, it'll have radiology to learn from, but we really are going to still need to be collecting the data sets with regards to gene expression, with regards to immune infiltrates, because artificial intelligence isn't going to be able to understand these things unless it has data to work from. And so, that's kind of still, for those of us who do translational research, that's like on us to kind of be able to collect those samples, to do the assays, to try and take as much from it as we can and have those data sets that are going to be available as artificial intelligence continues to get smarter.
Thank you so much, Doctor. It was wonderful. The answer was wonderful, and your presentation was also really amazing. It was I'm really honored to hear this presentation from you. So for the next question, I'll pass on the mic to Madhuri.
Thank you so much, Doctor, for this wonderful presentation. It was really interesting to learn a lot on the microenvironment and how complicated and dynamic this whole situation is as well. My question is actually based on some of the physiochemical properties we see of nanoparticles, with it also being a possible variant in helping the immune microenvironment. How can it help overcome the immune escape and tumor progression?
Yeah, I think there's so nanoparticles, I think, are going to be really quickly able to help us with some of these intratumoral therapies, like we were talking about before, like the TLR4 agonist that I showed. And I think that it's going to be able to help us package different things together so that they that those therapies work even better, like the TLR4 agonist. So you can package cytokines along with a TLR4 agonist, or so I think that's going to be kind of like the sort of like immediate short run benefit. And then I think in the long run, I think some of these nanoparticles are going to help package payloads that you're going to be able to deliver systemically through IVs and send things that would be toxic. Like, for example, I mentioned you can't just give a TLR4 agonist into the peripheral blood of a patient to treat all of the 100 metastases that are in a patient's lung, but you may be able to give it in like packaged within nanoparticles, right? In a way that it's going to be able to get the nanoparticles are going to home to the tumor and be able to release the payload there. So I think that is going to be something that is really going to change things.
So much, sir, for your presentation. I truly learned a lot from this session. So my question for you is, for our more junior attendees just getting into research and medicine, what inspired you to pursue this field of oncology, and how has this choice impacted your life? And is it everything that you had initially anticipated?
Yeah, so that's a great question. My mom died of breast cancer when before, like when I was a resident, and that, like, when I was kind of still kind of putting things together, and she was suffering from the cancer and the different treatments for many years before that. So I think based on that experience, that's really kind of what drove me into the field. In terms of the field of rare cancers and sarcomas, that came more from just like the science, and I got interested in that. When I was in school, or when I was earlier in my training, bone marrow transplant was like kind of a hot topic. It was very new, and today bone marrow transplant, it's like kind of old news, but that back then it seemed like it was something that was so high tech that there were patients being cured by taking cells from one person and putting them into another person, and it just seemed like it was like so fascinating that and you could kind of see how the science, it was very like clear how the science was leading to changing the patient outcome. So I got very interested in that, and that's kind of was my first sort of foray into research. Research is tough. I mean, it's most things don't work, right? And it can be very frustrating. Cancer is tough, right? Sometimes after, if you're treating patients, it can definitely wear on you. But I would say overall, it's wonderful to be able to be a part of it, right? It's very exciting, and I think it's very important, right? It's very meaningful when you do help patients. So I think if you're interested in cancer, if you're interested in research, you should go for it. You should go for it. And it's hard. So if it's not for you, some people kind of feel like maybe I should do, maybe it's not for me. And, if you're feeling some sort of like other push to go into it and it's not really in you, it's not going to go well, right? Because you have to get through those hard times, right? But if it's in you, you should do it because it's a really great thing to do.
That was such an inspiring news. Thank you so much.
Thank you so much. A huge thank you to our speaker for an amazing session, and thank you for all the great questions, and to all of our attendees. So with this, we come to an end to this session, and please continue to follow MedNews Week on all our platforms, and make sure to catch our
I just want to say one time,